Cool. I'm a second-year grad student in Ben's lab. And this is kind of how I'm approaching this problem. And I also want to end this talk eventually with how you guys are thinking about interpreting your data using machine learning. This is my system. I'm studying amoeboid cells. This is a bone marrow-derived macrophage from a 7-month-old mouse. And one of the things that I'm trying to do is I'm trying to take morphology, aka shape and dynamic, so morphodynamic data, and interpret that data, find structure in it. And hopefully that data will provide insight to cell state. So macrophages, immune cells. I'm hoping that basically I will be able to get this morphodynamic data to describe the pro-inflammatory or anti-inflammatory states of this cell. And I'm using machine learning tools to do this. And this is, it caught up. This is a quote from Tim Miller. He's out of the Center for AI and Digital Ethics in Melbourne. And it gets to the key of, I think, what you have to figure out when you're deciding what kind of machine learning models you want to employ when you're analyzing your data. Then this is interpretability of a model is the degree to which an observer can understand the cause of a decision. I think that this is incredibly important to consider when you're using machine learning for data analysis, because there are trade-offs between interpretability and precision, especially with the rise of opaque models like I'll talk about later, versus traditional models, which are inherently more interpretable. And so interpretable models are things like linear regression and principal component analysis, which are excellent at elucidating trends in data or structure in your data. And from these kinds of traditional models, because they're interpretable, we can identify key biological drivers that are creating these trends that we see, and we can flag those for future inspection. And this gets to a few different questions that we want to answer in biology, because we always have like a what's happening, but also a why and a how. And I think that these types of applications are really good at helping you figure out that why and the how, versus opaque models, which have seen a huge rise in use. These are specifically, I'm talking about deep learning models. And so the way that these work is that you put your data in, it goes through a number of analytical layers, and then you get a result out. And these are pretty bad at the why and the how, but they really give you a what. And sometimes that's incredibly the only thing you need. But sometimes elucidating those biological drivers are an incredibly important part of answering the question of of the biological significance of the thing that you're investigating. And so I wanted to talk a little bit about someone who is doing work in this field and trying to use these deep learning models to gain biological insight. And something that these opaque models are really good at are predictions. And so this is out of a paper from Margaret Gardell's lab, and I think she worked with Dr. Oates and Vitelli from U Chicago, and Margaret Gardell's lab, they study cell migration and adhesion. And so the goal of this particular study was to see adhesive proteins in fibroblast cells. They had fluorescent labels, took images of them, and then put those images through a deep learning network in order to see if the network could predict adhesive forces on the cell. And they found that it was incredibly efficient at being able to predict those forces. However, they explicitly noted that there was no transparency. And so their understanding of the biological drivers required a bunch of iterative testing and bottlenecks for this kind of training. And so I want to talk a little bit about how I'm using machine learning, both in this opaque way and also this transparent way, in order to elicit biological significance from my data. I put up a picture of some of my cells. I'm hoping that if you work with cells, some of you will know this is a now very common cell segmentation tool called CellPose. It's a deep learning model that I don't really need to know what the key biological drivers are here. I just need it to do this work for me, because this work is incredibly time extensive. And so I'm using this model in a way that I hope is going to help me find significance, which is that I don't need to know what's happening at the top, but I do need to know what's happening at the bottom. So I'm using this PCA, one of these traditional interpretable models, to look at my cells. This particular data is using synchronized macrophages, and again, taking the morphological extractions from the top and inputting it into my PCA. And because of that, I can interpret what this actually means. I'm going to describe it a little bit for you because I think it's kind of exciting. So these are all macrophages. They're all metabolically reset. And I have a time course of them. And I can see that they're actually incredibly polarized at different times. And to me, when I'm looking at this question of anti-inflammatory or pro-inflammatory, seeing this polarized morphodynamic action indicates to me that there is likely a pattern of morphodynamics that's following this cell state. And so that's how I'm using machine learning in order to predict cell state. And I would like to open it up for your guys' thoughts on appropriate uses of machine learning. Yeah.
Very cool. I don't know how to think about PCA and shape with the types of cells that you have.
Great question.
In
Okay.
particular, like your cells break all sorts of symmetries that like an eigenworm type analysis wouldn't break. So like how do you not sort of believe that there are actually more dimensions than there are under some symmetry?
Great question. There are a billion dimensions. There actually, this is a somewhat older model. This only uses 23 features, 22 dimensions. I'm currently working in over 600 features, so extremely high dimensional data. And PCA is breaking down what it thinks is the basic contributors from those. So currently I'm working with 37. This is my top two.
So the features, they're morphological features.
They're morphological, yeah, sorry. They're measured from here.
So, like shape descriptors.
Okay, and some of those shape descriptors are like rotationally invariant or something like that.
Yeah, so for this data, for example, I can tell you that PC1 is elongation. So cells over here are extremely circular and cells over here are long. And then I start to see the stratification with texture features. So like, I'm so sorry.
No, that's great.
Cells that have more texture and cells that have less texture. So that's how I'm breaking down this data. Other questions? No. Yes.
Find the variables that got fed into the...
I define them using just like a basic feature extraction. So they already exist.
Cell pose or...
No, I used a different package for feature extraction. Yes.
Dynamics?
I have used the dynamics. I'm not showing that data because it's not complete yet.
Say more.
What I can tell you is that I've created vector flows for this exact data, and it seems like there is a pattern of movement that's somewhat torus in shape.
And so we're most interested actually in flows in this feature space, and we think that those are really going to be what's diagnostic from these relatively straightforward, easily acquirable data sets for the cell state inference with
Okay.
limited underlying data. Yeah.
Any other questions? Okay. I'm going to leave you with a special thought, which is like CNNs or deep learning networks are really good at image data, like very specifically. So if you have image data, try it out, see how it works for you.
You.